Hi, good morning, and welcome to a new series called Breaking Through, which is a live discussion brought to you by Breakthrough Cancer. I'm Tyler Jacks. I serve as president of Breakthrough Cancer, and today I'm very pleased to be joined by two outstanding researchers who are members of one of the team labs that we have funded at Breakthrough Cancer, Doctors Nathalie Agar and Kenny Yu. I'm going to be having a discussion with them shortly, but let me start by asking them to introduce themselves. Nat, why don't you go first?
Yes, I'm Nathalie Agar. I'm Associate Professor of Neurosurgery and Radiology at Brigham and Women's Hospital, Dana-Farber, and Harvard Medical School doing research mainly on brain tumors. Delighted to be here.
Kenny?
My name's Kenny Yu. I'm one of the neurosurgeon scientists at Memorial Sloan Kettering. Nice to be here too.
So Nat and Kenny are members of a team that is studying one of the most challenging cancers that we face, glioblastoma, and we'll have an in-depth discussion about that disease and the work that they're doing to try to make progress against it. Before we do, I'd like to put our discussion in context and talk a little bit about Breakthrough Cancer itself. Breakthrough Cancer was formed about 4 years ago to tackle long-standing challenges in cancer. Cancer remains a devastating disease. There are 10 million cancer deaths in the world each year. In the United States, there are 600,000 lives lost to this disease. And although we have made progress as a field, there's still quite a lot of work to be done. I think probably everybody listening to us this morning has either faced this diagnosis themselves or is close to somebody who has. A diagnosis of cancer brings a range of emotions from fear and confusion, anger, and frustration. And it's actually that frustration that really drives us. We wish the progress were faster, and we're working in every way we can and in new ways, as we'll discuss, to accelerate progress against this disease. We were inspired to form Breakthrough Cancer by our founding donors, Bill Goodwin, Alice Goodwin, and their family, who have been supporting cancer research in the United States for decades now. And although they've been satisfied with the philanthropic investments that they have made to date, they too have been frustrated that we haven't made more progress. The Goodwins have supported outstanding cancer research centers in the country at MIT, at the Dana-Farber Cancer Institute, at Memorial Sloan Kettering in New York, at Johns Hopkins University, and at MD Anderson. And as I said, they were satisfied with the work that had been done with their support, but they actually challenged us, the leaders of these institutions, with the question, if you approach the problem differently, could you do even more? Could you go even faster? And specifically, if you worked collectively, if you joined forces, could you accelerate progress? Could you bring an even greater sense of urgency to the problem? And we leaders met and decided that indeed there were opportunities to think differently, to chart a new course for progress against challenging cancers, to form teams of investigators from across our institutions and indeed from other institutions who would form investigative groups that would think differently, think together, envision new projects collectively, commit to executing those projects as a team, to share data in real time, to share samples, to share credit, to approach the problem of cancer with a very patient-focused orientation, not about the next academic paper that they would publish, but about progress that they would make in changing patients' lives. This would create or would require new structures, new organizational structures, new ways of thinking. And Breakthrough Cancer was born to provide that infrastructure. The organization supports the formation of these teams of investigators, supports the execution of those projects, which are complex in nature, and you'll hear about an example of that today. We obviously monitor the progress of the teams, but we also help them overcome barriers that they encounter in their day-to-day work. We help broker relationships between the institutions. We involve companies where necessary to bring them into this process. We foster a new style of research which we have dubbed radical collaboration. And it will be this style of research that we'll focus our discussion on today. I mentioned that we'll be discussing glioblastoma, a very important cancer type. Breakthrough Cancer decided to focus on 4 cancers at our inception: glioblastoma, pancreas cancer, ovarian cancer, and acute myeloid leukemia. And these were chosen because they're hard, because progress against these particular cancers has been slow, and it's been decades since we've had real advances in any of them. We now collectively, over the last 3 years, have formed 9 teams of investigators, and they are working in a remarkably harmonious fashion. They are indeed functioning as though they were in the same laboratory in one location, despite the fact that they're located across the country. They are indeed sharing ideas and sharing data and sharing samples and executing a plan with our support that is very focused on advancement in the clinic to bring the best of science to the patient and to learn in the process as we go. Unlike traditional funders, we do not just give money. We at Breakthrough Cancer stay deeply involved. And as such, we get to hear about the progress that our teams make literally daily. And it's incredibly exciting, the clinical advances that they're making, the technological advances that they're making. And we decided to form this series, Breaking Through, to share those with you and to share our excitement with the large audience of individuals who are interested in progress against cancer. So we think that this will be an informative series. We hope you will learn from it, benefit from it, get excited about it, and share with us in the enthusiasm we have about what we can do as we think about progress against cancer in new ways. So now let's get into our discussion about glioblastoma. Glioblastoma is, as many of you know, a truly terrible disease. It's not the most common cancer that we study, but it's probably the most deadly. The statistics are that an individual who's diagnosed with this disease has only about a 5% chance of surviving after 5 years. That's very frustrating. I want to turn to Kenny first and ask him, from the point of view of a surgeon who takes care of patients with this disease, what's your perspective on glioblastoma and its challenges?
Yeah, thanks, Tyler. So, yeah, I mean, glioblastoma or GBM for short, really, it really is a devastating disease. And, anyone who's treated patients with GBM or even, patients and their families know that, it takes a big toll on the patient and the people who care for them. And the reason why it's a difficult disease is because it's not a tumor that can be easily resected or just cut out, because the way the tumor grows in a very vital organ, the brain, obviously, is that it infiltrates, into the brain. Somebody once described to me when I was training is that it's like dropping some paint into like a background of white paint, and it's just trying to pull that colored paint out. It's quite challenging, and so surgery alone is not the solution or the only solution. It comes as part of like a package of care that we try to deliver for our patients. But even then, the problem and the challenge is that oftentimes it comes back, nearly always comes back. As Tyler mentioned, there's only a 5% chance of long-term survival. But also the fact that there is a chance of a long-term survival means that some treatments probably do work, and we have no idea why they work. And so I think that really motivates us, I think, as clinicians, as researchers, trying to figure out, what can we do? What can we do better? What doesn't work? So that's really, I think, some of the premise of the work that we're doing right now.
Yeah. Nat, you've been involved in this area of research for a long time. You've seen attempts to develop new therapies for glioblastoma. Talk a bit about that history. How's it gone? Where are we today?
Yes, so I think it's not for lack of trying. There are a lot of studies that have been going on. Sometimes we'll launch into clinical trials with some promising preclinical data and certainly a lot of hope, but really standard of care for GBM has not changed over 20 years. And the way that the clinical trials are designed is that we look for overall survival. And as Kenny just mentioned, like there is signal somewhere, but we're missing that signal. So effectively, over a period of 10 years, we've had maybe 8,000 patients on clinical trials, and every single one of those trials has failed. But the way that we do things is that we just stop there and run to the next exciting target and do not learn from those clinical trials. And so this is essentially what we want to be able to do now, is to learn from clinical trials.
Yeah, I remember those early discussions as we brought the group of investigators, which eventually formed this team together now 4 years ago. That statistic that you just referenced, 3 decades of failed phase 3 clinical trials, that was rather striking. So you gathered, the two of you and others across the institutions that I mentioned previously. Talk about the origin story of the glioblastoma TeamLab. How did you come together? How did you decide what to do together? And what were some of the experiences and even some of the challenges that you faced as you put that team together and began to execute this project? Nat, why don't you start?
Yeah, so I've been extremely collaborative my whole career, and but this one came very differently, right? Is that essentially Breakthrough Cancer had identified institutions. Each institutions had named some leads at each of the site to bring a team together, and essentially brought onto many Zoom calls, many hours a week, essentially, to come together both around ideas, but I do think coming together as a team. And so that was both challenging and inspiring. And I have to say, for me, at some points in time, I thought maybe it was a big risk to my career. But on the other hand, I could not give up. It brought a lot of hope, because then we had the opportunity to do something that was so much bigger than what any of us could do individually. And when I say individually, it's even each of our laboratories. So basically, I stuck with it, and I'm definitely glad that I did. I must say that I don't think it's a model for everybody, but I've learned tremendously, and I think what it gave also is that flexibility, so that people have come and gone in part of those discussions, and they kind of dialed in and were part of some discussions, and it was not for them. They decided to go, but the product is a team that grew organically together with that same drive to come together as a team to make a difference for the patient, not for each of our own um, essentially career development, but challenging but extremely inspiring. And I must say that the support that Breakthrough Cancer provided in that process has been extremely rewarding, but also educational. I've learned a ton doing it.
Yeah, just to pick up on something that you said, I think we are a collaborative community in cancer research. Many of us work together, but this is a different style of collaboration, depth of collaboration, which is unusual. And in many cases, and I think it's true of your team, competitors become collaborators, competitors become colleagues, which is interesting and unfamiliar sometimes. And maybe especially so for a younger person, a junior investigator earlier in their career. And Kenny, you're sort of in that spot. I won't mention your age. I actually don't know your age, but talk about this from the perspective of somebody who's at an earlier point in their career.
Yeah, so I came into this the process sort of later on. I think there were a lot of previous discussions in terms of the way that the project was going to form and evolve. But I was brought in because, like Dr. Sabar very kindly said, well, they're talking about some of these techniques that you might be familiar with. Maybe you should just join in and see what it's about. And I remember attending these Zoom meetings, and it was actually a little bit intimidating, to be honest, because in this meeting were basically all the luminaries in the field, all the big names that you've been kind of reading about your whole career, basically, and suddenly you're shoved in the room, and I just sort of thought, what am I doing here? Like, but then I think through as the project sort of slowly started to come together, And you know, a lot of these questions about like the nuances or or just the technical details of how we do stuff like came about. And I thought, actually, you know, I've done a bit of that. Maybe I could answer some of those questions. And what really I felt, and I honestly felt was really inspiring for me, is that, you know, they didn't treat me as like just like an outsider because I was junior and nobody knew who I was, but they just sort of said, actually, you know, everybody's very welcoming. And I that's, to be honest, what kind of inspired me to like keep going on and say, actually, maybe I can contribute something to help this project. So yeah, that's kind of how it started really for me.
Yeah. Yeah, and it's been a tremendous amount of work by all of you. I remember the number of meetings that you all had in planning this was remarkable. These are busy people. I remember there were meetings that occurred at 7 or 8 a.m. on Tuesdays every week to talk about what you were going to do, not even what you were doing, but rather the planning phase. And that's continued throughout the course of this project. Nat, you mentioned that you're prioritizing the project, prioritizing project progress, and there's a certain selflessness to that. There's sort of a recognition that it's more important to think about what's going to happen for those patients than rather for your career. I think that's a really important point for people to understand. We are asking our investigators to behave differently, and really, it's not for everybody. This is not a style of science that fits everybody's style, and that's not a bad thing. But we need to find folks who, like you two, are committed to taking on this approach. So let's talk a little bit about what you're actually doing. So this is a clinical trial supported by a tremendous amount of science. I'd like to talk a little bit about the clinical trial, the agent, and then we'll get into the science and how it's differentiated. So Nat, do you want to take that on first?
Yes, so our team came together wanting to learn from our patients on clinical trial and wanted to see if we could take biopsies from the brain during treatment, which really had never been done before, but it's a concept that is done in other types of cancer. The idea of biopsying the brain was thought to maybe not be well received in the field and for patients as well. But then the surgeons on our team felt that, the tools and the technologies that they have to do this has made it so that now biopsies are safe. And so we wanted to develop a concept for a clinical trial that would also to essentially balance the potential risks and benefit is that, well, if we're going to go into the tumor, we would want to be able to deliver a treatment, so potentially benefits by going into the tumor. So we based our clinical study on an immunoactivator, a virus essentially that we are injecting into the brain. This was a virus developed by Dr. Kioka on our team, and for which he had already a lot of clinical data in terms of safety and so on. So we this is essentially the platform that we use now to validate the safety, the feasibility, and the value of doing biopsy for investigative purposes, essentially in the brain for patients on trial. And at the same time, we've been collecting or aim to collect imaging and blood and CSF to see if there are signals in those other compartments that we could potentially even use that would mirror what is happening into the tumor.
Yeah. So a very deep, scientifically driven investigation of what happens when a patient is treated, in this case with an oncolytic virus, in ways that in brain cancer at least have never been done before. Kenny, as you heard about this concept being a neurosurgeon, what was your reaction to the feasibility that patients would sign up for multiple biopsies over the course of weeks, actually months of treatment, or that institutional review boards who monitor, the safety and feasibility of studies would agree that this is something that was justifiable? So how did you see that?
Yeah, I mean, so stereotactic biopsies like needle core biopsies are something that we do quite reasonably often. But typically it's really only for diagnosis, right? It's like the idea of doing an extra biopsy, if you like, for non-diagnostic means is not common. Every now and again, if there is some diagnostic uncertainty, then you might think about doing another biopsy. But to do it outside of this realm is a little bit, off-piste, if you like. The so I think initially when this proposal was discussed, there's a lot of concern about, well, what's the safety of doing multiple biopsies? We know the safety of biopsies in general is really quite safe, but what about doing it if you do it multiple times? That was the big kind of unknown. So I think the fact that we had a framework for a trial where we're already injecting something in, so you have to go in anyway. But basically piggybacking on that framework to say, well, while we're there, why don't we take a sample from the tumor so that we can actually see what's happened in the interval? I think that made the justification a lot easier. Because it's not like we're doing a completely separate operation. It's really in the same operation that they're doing both things. So that made it much more palatable, I think, in terms of like getting through IRB or that kind of thing. And that really laid the groundwork for all the scientific aspect of it. And I'll just also mention that throughout all the discussions, it was the idea was, yes, let's do all these different fancy kind of techniques, but the amount of tissue you get from these needle core biopsies is tiny. And for us, we only really look at or the pathologists really look at H&E and some basic molecular testing and stains and things. We really weren't sure whether we can get anything more than that. And we're basing a whole trial on the idea that we're going to take lots of samples, do fancy things with it. But that's what a lot of the preliminary discussions were. Well, can we even do this, physically, technically? So,
yeah, I want to come to that because I think it's interesting, and we'll come to what you're learning as well. But did the patients sign up? There was uncertainty. Would patients actually enroll in a trial like this that required really so much of them? What was your experience? Kenny, why don't you take this too? What was your experience? What was the group's experience in patient interest?
So really surprisingly, from the patient side of things, they were actually very eager. In many ways, they're very keen. Because there's lots of trials. Well, when the disease comes back, and the fact that this is a kind of innovative treatment anyway, and you'd be able to get additional information at the same time. I think it actually appealed to patients quite a bit. And I think the way that some of the groups did it as well, it's almost like in fact, it more or less was a day case as well. So, the actual burden in terms of hospitalization and things was actually relatively low. So from that side of things, it's been pleasantly surprising how well received it's been from the patient's perspective, I think.
Yeah, and I too have been impressed. And one thing I've learned in sort of watching this and other trials is the willingness of patients to be participants, to be partners. When I talked about radical collaboration earlier, I include the patient in the context of radical collaboration because they are critical partners in this learning system that we're building here. Obviously, the trials are designed to provide benefit to the patients, but they're also designed to learn. It seems to me that among the reasons why there's been failure in glioblastoma, and there's more than one, is that there's been a failure to look. If you don't look, you cannot see. And I think there's been a failure to look deeply. And with the tools that we have at hand today, we can look very deeply. And this trial is an example of how to do that and may actually serve as a framework for trials for GBM into the future. So Nat, Kenny talked about these tiny biopsies, and there are many per patient per time point. Tiny biopsies. You guys thought a lot about the logistics of that, the feasibility of that. Could you get data? So speak, if you would, about the feasibility question, sending these tiny samples around the country for analysis, and also whether indeed you can learn interesting things from what you get from these small samples.
Indeed, we did talk a lot about this and how to organize ourselves to make it happen. And as Kenny mentioned, one of the questions was those specimens are so tiny, are we going to be able to learn anything from them? So we didn't jump into this with our eyes closed. We did a careful study led by Kenny here and other colleagues on the team, where we essentially mimicked the trial in a sense that tumor samples were taken out, but then biopsied as well. And then we tested, optimized essentially all of our platforms or assays to be able to do this on those small samples, but also did a test drive of our system of sharing specimens and then sharing data, and not just sharing them, but integrating that that data. And we were quite surprised of the quality of data that we are able to get from those tiny samples. But that was and I should let Kenny talk more to this, because that was a lot of work in the lab by many teams working together to essentially optimize those assays. The other thing that we've done, one question was about heterogeneity. Biopsies are those teeny tiny samples representing a whole tumor. So we'd want to know from one location to another whether they're comparable or even within one location. So we also developed tools to be able to essentially measure this. Everybody says this disease is heterogeneous, but we don't have a way to evaluate that heterogeneity. And then again, the team here did something along those lines, where now not only do we have the assays, we know what we can compare to what within a tumor, and if we can integrate this this data, which led to a manuscript that has since been published, just on, we called it a pilot study essentially.
Yeah, and that did demonstrate the feasibility of this approach, and that led, I think, nicely into the clinical trial that is ongoing. It's a small scale trial. 12 patients will be enrolled in this trial. We're in the middle of it. Those patients have
Past
been
the middle, Tyler.
past the middle now. Happy to hear that.
Yes.
And many samples have been retrieved and analyzed. I'd like you both to comment without going into detail that you can't share because it is an ongoing study. But what are you starting to learn? That was part of the goal here, is to learn more in the course of the study. So give us a sense for what you're learning and why this depth of analysis actually is beneficial in how we evaluate what's happening in the brain of a patient undergoing treatment.
Yeah, so I think one of the things that was in some ways, I think it's almost like an anchoring technology was like, for example, was like the single cell RNA sequencing for these biopsies. I think one of the early discussions that we all had was everybody wanted to try and do single cell sequencing on the biopsies, but it just wasn't clear whether we could actually do it because all the previous studies had been mainly on resected tissues, and even in the some of the biopsy studies which actually tried to do single cell, the number of cells that was actually extracted was very, very low. So we spent quite a lot of time optimizing these different protocols. And again, this speaks to the collaborative nature because everybody just shared their protocols and sort of said, well, which one should we use? And so we ended up settling on, well, let's do a shootout, right? Let's just institution one provides the protocol, institution two actually applies it as though they were doing it for the first time, and then just testing out various protocols in a sort of like an unbiased manner, if you like, to see which one gives us a higher yield. And actually that worked very well, and everybody starts to also get comfortable with the logistics of sample transport and all these sorts of things. And then the second layer, which I think Nathalie alludes to, was we were kind of frankly surprised at how much data we could generate from these things. And so the next question was, well, can we actually try and link these different data modalities? The integrated process actually became a whole new level of like thinking, if you like. And this is also where the collaborative team comes into play, because everybody has their own kind of areas of expertise, but kind of cross-pollinating these areas of expertise was actually super fun for me, actually. Like Nathalie mentioned the heterogeneity study, it was like working with the spatial people, working with the people who are experts in proteomics, immunometadonics. I mean, there's so many really talented people in this team that gave us the ability to start to put these different strands of data together. And I thought that was something that was really exciting.
Yeah, it is exciting to watch even from our side as the funder here, the dynamic that exists within your team and other teams as well. Good ideas come from different places. Expertise lives in different places, and often the best ideas come at the intersection that you didn't necessarily expect. So we've seen that play out here. So Nat, talk a bit more about the data as you've seen it. Again, these are patients who are undergoing study with an oncolytic virus. You can look at gene expression changes by single cell, changes in T cell abundance and clonality, immunopeptidomics, and many other features of what's happening in the brain. Give us a flavor for the sorts of things that you're starting to learn.
Well, if we take a step back, one thing we've learned so far is that this is feasible. We can do these types of studies in our patients. And one point that we didn't bring up yet is like typically in clinical trials, like the endpoint is very much an overall survival, and the way to monitor the patient is based on MRI. And what we've learned already so far and we've presented some of this data already at ASCO, is that if we had been judging whether the patients are progressing or not, or responding or not, just based on the traditional ways of monitoring the disease, we would have been fooled, essentially. We would have thought that the therapeutic is not effective, probably stop, and then the patient moves on to another clinical trial. What we've learned here is that when we take those samples in the brain, we know exactly where they come from based on the preoperative imaging. And then we do the analysis on those samples. And what we've seen now is that there really is, and people in the field have been suspecting this, and there has been evidence of this, but it is clear that there is a disconnect, and we should not just be relying on the MRI to monitor response to the treatment. And so in a nutshell, Dr. Kioka's team had already shown, promising results with the virus, and the idea of this virus is to recruit an immune infiltrate to the tumor. And basically, those molecular analyses that we're doing now, including RNA analyses, protein analyses, and histology analyses, show that in fact the virus is recruiting an immune response to the tumor. And now the team is working really hard, and it's extremely rewarding to do this. We are also learning about the dynamic of that response, and it is extremely exciting to start, pulling these data together to understand in more details what is it doing. So some of it was known to a certain extent. Now we can confirm it, but now we're in the next level of learning, which is both exciting and extremely promising.
I would also just jump on to the idea that we're using MRIs like to really as a surrogate for disease like progression. And clinically, at tumor boards pretty much every week, the question is invariably we look at the MRI scans as well. Is this progression? Is it something else? Is it a treatment effect? And there's actually limited information that we can derive from these imaging modalities, and to actually have the underlying molecular data, like while the disease is essentially like evolving under treatment, is just it's incredible insight into the disease process itself, actually.
Yeah. I certainly agree with both of you, really. And I just to repeat myself, you don't see if you don't look. The brain is a complicated place. The treatment induces changes. They're remarkable, these changes. I've seen some of the data, the changes that are taking place in the cancer cells, the changes that are taking place in the surrounding so-called microenvironment, in particular with respect to the immune cells in their microenvironment. There's a lot happening there. In some cases, we hope that what's happening will actually lead to impressive responses in the patients. In some cases, it may not, but by looking, we'll begin to understand perhaps why not,
Exactly.
and what we can do next time to improve the outcomes with real data and not just suggestions or hypotheses based on not the real thing. So it's remarkable. This learning system is paying off. Nat, I want to turn to something that you said in passing. The biopsy method, which is the focus of this trial, works well in this setting in that you're injecting the virus and thus you're there already. You can take additional biopsies for study. We can imagine a time when the treatment might not be an injection into the tumor, might even be a systemic injection, and perhaps we would need alternative ways of molecularly following the disease and the response to treatment. So you mentioned cerebral spinal fluid as a surrogate, or even blood as a surrogate for monitoring the response to treatment. Talk a bit more about what is CSF and how it can be useful. And indeed, in the early days, what are you seeing? What is the team seeing as you look in the cerebral spinal fluid? That for the audience, that's the fluid that sort of bathes the brain, and material can make its way into that and be a reflection of what's happening in the brain tissue itself. Go ahead, Nat.
Exactly. Well, I think I'd like to break my answer into two aspects, Tyler, because yes, it is invasive to do biopsy in a way, and we thought maybe we would need multiple clinical trials with an intratumoral injection of an agent to further validate the concept. But so far, the team has become extremely comfortable that we could even potentially do this when we're doing systemic delivery of an agent. So it would still be, those are concepts that we're considering now, even for future clinical trials, as we're developing this as a platform, not just one clinical trial. But again, we want to learn from each of those. I think it will very much also depend on what the mechanism of action of each of those therapeutics is, to see how much of those other compartments in the CSF and in the blood, what are the signals that are mimicking what is happening into the tumor. So again, so far, I have to be careful what we're discussing here because this is ongoing, but we have been performing, and our colleagues at MSK and at Johns Hopkins are doing the analysis specifically on the CSF and seeing how do they correspond to the signals that we're seeing in the tumor in the sense of building that immune infiltration. And there is hope that we would be able to, have, there's something to work from. This is what I guess I should be saying here. And the idea is that we could do eventually rely on those compartments, again, depending on the therapeutics, or perhaps do a combination and of sometimes doing a biopsy or CSF or blood. But right now we're doing everything and learning from what each of those systems is telling us to see how to best use them. One other aspect of this study, one thing that we had said we would want to do is to see what is it that we're learning from the patients in a clinical trial that would inform perhaps doing a combination. So the hope would be that if you're learning from the patient and all of a sudden you're starting to see a pattern of failure, but you understand what that failure may be, then how is it that we could start already thinking about enhancing that response by bringing in something else? We're not there yet, but it's something that we want to be able to do.
Yeah. It's very powerful. It's still ongoing. But again, the hope would be that by doing the comprehensive analysis that allows you to see the tissue on the one hand and the CSF and blood at the same time, you'll be able to correlate changes that you see in one setting with others, and that may turn out to be a useful surrogate for future studies. So let's talk a bit about, I want to touch on two other things, and then we'll open it up for questions from the audience. Kenny, I want to turn to you. This project has generated a tremendous amount of data. We've talked about many biopsies from many patients over time, many types of analysis. That generates a lot of data. You've been deep in the middle of that. Breakthrough Cancer has emphasized real-time data sharing, the ability of investigators within the team to see data as they're being generated, to learn from those data, analyze those data. Talk about the data science challenge, and if you would, the value of the data science team and the data science hub that Breakthrough Cancer has established that's helped your team be successful.
Yeah, I think I think all of us were a little bit taken aback at just how much data can be generated from something like this. And it became apparent, actually, even from the inception, the data science team was already kind of preparing for this. I think it really helped that many people on the data science team, like Mike Noble and things, had previous experience from the TCGA, and so they brought in the same kind of like framework or structure that they did for that kind of big multi-institutional project to this study. And while the data was being generated, obviously people start like having ideas of how to play with the data, and sort of as a side data science group actually, which is almost what is a parallel team within the BDC family, if you like. They arranged like hackathons and things, and again, the beauty of those is that you get to meet people from other teams who actually are often working on very similar problems. It's not like spatial transcriptomics in GBM is completely different from that in pancreas, right? We're still trying to find niches, trying to find interaction, things like that. So like there's a lot of cross-pollination in terms of like techniques and like expertise. And again, having the data science program working closely in parallel with us actually and actually, a lot of the pipelines, like the standardizations, are something as silly as just like, how are we going to call these samples? Is there like a universal nomenclature that we have to use so that everybody knows what we're talking about? All these sorts of things have to be worked out. And that's something that, has been, really important as well for us.
Yeah. I think just for the sake of the audience, as I mentioned earlier, we have multiple disease team labs that have approached different cancers in their own way, but there's overlap, as Kenny just mentioned, and there's opportunity for shared learning. And we bring folks together specifically for that purpose, which again, I think contributes to the acceleration of progress. We've also invested heavily in data science, and it takes two forms. We have an internal investment at Breakthrough Cancer, a data science group within Breakthrough Cancer run by Mike Noble that Kenny mentioned. And they interact with a larger group of people called the larger data science team that has investigators from the different institutions that are partners of Breakthrough Cancer. And collectively, that's a very powerful group that's doing science in their own right, but also supporting the science of the disease-oriented teams in a really, really productive way. So let's talk about this as a platform. We're getting started with the oncolytic virus and learning as we go. But I think the larger scale, longer term hope and expectation is that this will prove to be a new way of conducting clinical trials in GBM. And maybe we can replace failure with success over time. So talk about this as a platform and in your view, what the future looks like.
Yes, so that is certainly the hope that we will be able to have better treatment for our patients. So yes, we started with this one concept, but we always knew, and in fact, one of the aims that we had was to essentially create a movement within the community, within the field. It's not just about our Breakthrough Cancer team. So we are thinking now about the next agents that we're bringing into the platform. Again, that each of those agents would be teaching us something different, both on how we can do this and what we can learn from the agents, but also broadening our network, essentially, of like-minded people, like-minded team, working with pharmaceutical company, in terms of creating this movement and essentially change that we do things in testing drug for brain tumor patients.
Talk about this serving as an incentive for industry to this problem. It's not that there aren't companies that are interested in glioblastoma. There certainly are. Breakthrough Cancer is working with some of them and thinking about others in the future. But I would say it's not been a hotbed of activity for big pharma. So how could this approach change the view of getting into glioblastoma as a disease area for industry in the future?
Yes, well, and rightly so that there hasn't been maybe that much interest in pharmaceutical companies for this, is if you look at the track record of successes in that space, maybe not have been seen as a good investment. But again, now if we can learn from what a therapeutic does, I firmly believe we do have a lot of good therapeutics. We just don't know how they work. So our idea here is that partnering with industry, we could have smaller cohorts of patients from whom we learn what the drug is doing and what the limitations are. And I think we can have better informed later stage, essentially clinical trials, or be more nimble so that it essentially de-risks the investment from a company to invest in that space and to have essentially an intelligent development of those drugs for those complex, essentially tumors. So
Yeah.
we do think that even though the first study that we've done might look fairly expensive, right? We did just one agent and so on, but we do think that even financially, this is a small investment to learn and to avoid doing a clinical trial that is deemed for failure. And it's not just financial. At this point, I don't know that it remains ethical to do clinical trials without learning from our patient, like subjecting them to agents, even though they're promising in a dish, they're promising in a preclinical model. We don't have a good track record of long-term effect. So I do think I do think it will be appealing for industry
Yeah.
to
I
do
think,
these types of studies.
yeah, this is part of the bet that we're making at Breakthrough Cancer, that by investment, and I want to reflect back on my earlier comments. This is an investment made by a family, the Goodwin family. It's an investment made by individuals who care about progress against cancer. Pretty impressive to me. The bet is that if you apply the best of science in the context of clinical investigation, you can learn, and by learning, you can do better the next time if that indeed is necessary. I think that investment is necessary indeed to make progress. Industry tends to get to clinical trials and then simply ask the question, did it work or not work? There's much less in the way of deep investigation. Academic investigators with standard funding models typically don't have the funds to go deep in the way that we're talking about today. So we fill that gap. And I think by filling that gap, we hopefully will inspire this style of science and level of investment by industry partners. And I think we'll continue to work with them and help them realize that opportunity. In terms of your experiences, I want to sort of bring our discussion to a close to talk about a few other topics. In terms of your experiences, sort of looking back to the beginning, to those early days when you were young and naive, getting together with this group of investigators, some of whom you knew, some of whom you didn't know very well. Now you're 3+ years into this experience. Just give us some personal reflections on how it's gone, what's exciting, what was challenging, what's rewarding. What would be your advice to other investigators thinking about joining a team like this? Lessons learned and so forth. It's a bit of an open-ended question, but Nat, why don't you take it first?
It's been extremely rewarding. I do think I feel much stronger as a researcher in this field, knowing that we have a team, and that is a team that spans so much. Basically, we do have all of the expertise and the right likely-minded people to make this happen. One thing that has happened over the years is that we kind of lost even identity a little bit with our own institution. So we're all on the same team, and we laughed when we realized that we were not even sure whose institution somebody was from anymore, because we all identified as being part of this team. So it's definitely created a completely different environment, opened up the possibilities of what we can do for this disease.
Kenny, how about you?
Yeah, I mean, obviously I echo, everything that Nathalie just said, but I think the term kind of team science gets bandied around a lot when it comes to like, funding proposals, that kind of stuff. But actually executing true team science is actually not that easy, but if it can be done, and I think we've, in many ways we are doing that, is very rewarding, especially if the motivation and the question is interesting and exciting, right? And I've certainly learned so much just from working through all these wonderful colleagues. Actually, it's also spawned a lot of kind of like side projects as well, because that, now we're so familiar with each other. There's something else going on. It's like, well, you look at this, what do you think of this? So I think it's just had second order effects as well, I think, which has just been great, I think.
Yeah. So let me turn to some questions from the audience, and then I'll bring it to a close in a few moments. I started off by talking about the statistics, 5-year survival rate of 5%. I realize that we're in a small scale clinical trial, and what you're learning is not going to change those statistics overnight. But a question here is what you're learning going to make a difference in that 5-year mark? Do you expect that through this study or through the learnings from this study, you will change those statistics? That's a hard one to answer, and I appreciate that, but I'm curious about both of your points of view.
Within this particular clinical trial, you're thinking,
Well,
Tyler? Or is it...
as you think about the data that you're generating, this is not just science for science sake. This is science with the goal of changing outcomes. So, reflect on your sort of level of optimism. And I don't want to put you in an odd spot, but
Yeah. I think for me, I'm a very pragmatic person. The way that this allows us to do is I think we've had this idea that there might be almost like a unicorn treatment, right? Let's try this. It's worked well for breast or for lung or for, and then try it, hoping that it would make the big difference, and it has not. As a pragmatic person, what I'm seeing right now is that we can make adjustments and we can inch our way towards moving that needle here. And essentially, yeah, one aspect of the tumor at a time, we can start addressing it. And I do hope that this does lead. I am confident 5 years is so short, but I do think that already within 5 years, I mean, it's been 4 years, the things we've learned are applicable. I do think, I mean, I'm not a physician, but I speak, some of the physicians in the group seem to indicate that they are seeing their MRI differently, and that so yes, I do think we can have this kind of impact.
yeah. Kenny, what's your take?
Yeah, I mean, obviously, with any kind of data set, especially in terms of survival, there's always like a curve, right? And what I think we're trying to do is just trying to inch towards the longer side of the survival. And the more we understand, the more we can try and push this curve to the right. In terms of the 5-year kind of like horizon timescale, I think it's still too early to say, whether this is going to make how much of an impact like the current things that we're finding are going to make in a 5-year horizon. But I think these things snowball. I think if we can do more trials on different agents, we'll learn different things and different mechanisms, hopefully, as to how the disease adapts and becomes resistant. And as we learn more of these things, I think we're going to see an acceleration, hopefully, of that curve towards the right.
Yeah. The way I think about it and sort of reflected in your comments is to date, the trials that have been done based on real science and decent or good hypotheses were a little bit a wander in the dark. So it really wasn't enough information coming back from the trials to really know. And I think what you're doing, and I hope the framework that you're developing, is providing a flashlight or a torch to allow you to see your way through the dark and make decisions that are based on real information. This is what it actually looks like when we do this. Here's a set of molecules or cell types that we might now want to think about manipulating. And I think in the aggregate and in time, that will indeed lead to the outcomes that we're looking for, where that 5-year survival rate will change and obviously will improve. There was a question, it's a focused question on IDH mutations. For those of you in the audience who don't know, this is a mutation that occurs in a subtype of glioblastomas. It's an enzyme that's involved in metabolism, actually. I can't remember actually whether your trial focuses on IDH mutant or not mutant, and how that might impact how you think about what you're doing.
So
Kenny?
yeah, so IDH so our trials focus on IDH wild type. Um, and to answer the question in terms of whether the longer survivors had IDH mutant, the data came out more or less in the 2010s from the TCGA data, and that's when we started really stratifying patients on the basis of this mutation. Having this mutation definitely gives you a better prognosis to the extent now we essentially treat it like two different categories of disease. It's in fact no longer even called the GBM anymore for IDH mutant. They call it astrocytoma grade 4 or other things. So we're dealing with GBM, which is the IDH wild type component, which is the more aggressive subtype. But even then, we see people who have long survivors. So that's why there's still a distribution that goes on for these patients. Yeah.
Thank you. That was helpful. Um, I want to talk a little bit more about the future, and then we'll bring it to a close. At Breakthrough Cancer, as I mentioned, we're funding a number of disease team labs across very many important cancers. Glioblastoma is certainly among the most challenging. The team has made excellent progress in the clinical trial that we've talked about over the last several minutes, hour. And we're hopeful that this will serve as a foundation, a new model for pursuing clinical trials in this disease. We're actually seeking partners to work with us to form a funding coalition to support this style of research. And the team is thinking about new approaches, new trial types. I'll invite both of you to talk a little bit about that briefly. How do you see the future from the perspective of new kinds of things that you could do, not necessarily pinning yourself down to a particular detailed plan, but the kinds of things that you can do? We start with an oncolytic virus. What else might we do as we move forward?
I think the way that we've set up like our first clinical trial, we've definitely developed a lot of expertise in better understanding agents in the space of immunoactivation and developing the assays accordingly. But it doesn't necessarily mean that's all we have to do. Some of the agents we're looking at now for the next studies are very much within that space. With that said, it's a framework that should apply or hopefully will apply to much broader classes of molecules. But to be able to do this, to your point, I was reflecting back on the 5-year thing, Tyler, and like our team has been big, 5 institutions so far and growing. But I think that if we come together as a movement, yes, absolutely. I think within 5 years, we would have learned a lot collectively. So this idea of having a coalition or just even, having colleagues at other centers work with us along with similar ideas or apply them on their own studies, I do think is where how we can have an impact, and to share some of that data, to share those findings, to not think about it as being my own research project, but rather something that we collectively do for the patient. That's one big difference here. I'm humbled by how many hours busy physicians are putting into this, and that is the patients are driving them. That's very clear to me.
Eddie?
Yeah, and I would say that, the whole idea of the platform is almost like therapy agnostic. It's like it doesn't matter so much what the therapy is, it's what do we detect once we've given this? And actually this kind of answers the question that was posed in the chat as well. We do see some histological changes which may or may not be therapy specific. And so that's again, we're learning, and we would never know that if, we didn't see these changes over the course of a trial. And so I think, it's very exciting, I think, as we bring in more and more different agents into this platform.
Great. So we're going to wrap things up here. I want to thank Kenny and Nat for a really interesting and provocative discussion. Glioblastoma remains a difficult problem, but I have more confidence today in hearing about what they're doing, what their team is doing. And I hope you in the audience have learned a bit about Breakthrough Cancer and the approach that we're taking, radical collaboration, to bring people together from different domains, different institutions, to think differently, to act differently, to prioritize the patient and urgency for progress. I think this will indeed lead to benefits to patients and to science in ways that we can imagine and in ways that we can't imagine. So I invite you to stay tuned for future episodes of Breaking Through. Thanks for taking the time today. Take care.